Hello, my name is Soraya Ko. I'm a first-year master's student in the Department of Biology at UNCG, and I work in Dr. Bhandari's lab. Today, I will be answering the question, are grandparents' lifestyles responsible for their grandchildren's health? As your grandparents were growing up between the 1950s and 1960s, they interacted with thousands of manufactured items every day. However, unbeknownst to them, many of these products contained a chemical called bisphenol A, also known as BPA. BPA was often found in items such as food packaging, plastic bottles, canned food, cars, menstrual products, and even children's toys. We now know that exposure to BPA can lead to multiple health defects, including mutations to our epigenome. The epigenome sits right above the DNA within our cells. It reads out the information that DNA provides and decides which genes that the DNA codes for will be turned on or off. However, if the epigenome is exposed to certain chemicals, then mutations can occur on it that will change which genes are turned on and which genes are turned off. This is called an epimutation. This on and off itself is already concerning as it relates to the onset of diseases. However, studies have found that specific chemicals can cause these epimutations to occur on the egg or sperm cells of individuals. This means that a chemical like BPA could make changes to an individual's epigenome that could be passed down to their children, who could pass it on to their children, and so on. This means the effects of the chemical are transgenerational. The negative effects of BPA and its potential to cause heritable epimutations has already been extensively studied during the past 2 decades, and because of this, its use in products has become restricted. However, BPA is only one of millions of chemicals that individuals are interacting with on a daily basis. One new chemical group whose use has become environmentally prevalent within the past 2 decades is perfluoroalkyl substances, also known as PFAS or PFAS. These chemicals have oil and water-resistant properties. This means that they have been used in the production of items such as nonstick cookware, fast food packaging, stain and water-resistant items, firefighting foams, and paints. This continuous use has caused PFAS chemicals to accumulate in the environment, leading to even further exposure and chemical buildup within individuals. In 2018, it was found that 99% of Americans, including newborns, have PFAS in their bloodstream. Unfortunately, our nearly ubiquitous contact with these chemicals has been linked to numerous adverse health effects that can include a decrease in vaccine effectiveness, as well as defects in thyroid regulation, reproduction, early development, immune response, and liver health, in addition to an increase in the prevalence of certain types of cancers. Considering this, our most recent generation, born between 1997 and 2012, are currently carrying BPA-specific transgenerational epimutations that they inherited from their grandparents. However, they are also the first recorded generation to be born with PFAS already in their body, in addition to continuous exposure post-birth as well. This unique situation has not been thoroughly explored. Therefore, I'm working to discover if the current generation with a history of ancestral BPA exposure have developed an increase in sensitivity to the negative health effects from exposure to emerging chemicals of concern, such as PFAS. In order to answer this question, we use the chemical PFOS, which is a member of the PFAS family. We also used Medaka fish to mirror human history of exposure. The reason Medaka fish can be used to look at human responses to exposure is because there are 40 regions of related gene content between Medaka and humans, and both process the epigenome in egg and sperm cells in the same way. In my experiment, 2 groups of embryos were used. Group 1 did have a history of ancestral BPA exposure, and Group 2 did not. Both groups were exposed to 5 concentrations of PFOS. Embryos were exposed from 8 hours post-fertilization until 15 days post-fertilization. During this time period, stem cells are developing into specific types of cells, such as liver, brain, bone, or heart cells. After day 15, half the fry used were transferred into clean water to continue to grow. This procedure was repeated 3 times to ensure the accuracy of results. The rationale of this experiment is to see if the effects of PFOS on early development of embryos who have or have not had a history of ancestral BPA exposure. We examined the effects of the lowest concentration of PFOS, 0.002 milligrams per liter. This concentration is essential to examine as it reflects the same level of exposure we would expect to find in the natural environment. we found that during direct exposure period to PFOS from day 1 to 15, the individuals with no BPA epimutations had a lower rate of survival in the 0.002 milligram per liter concentration. Therefore, we can conclude that early development and direct exposure from PFOS results from lower mortality rates in individuals with BPA epimutations compared to those without. In contrast to this, we found that when looking at the survival of fish after direct exposure from days 15 through 35, when they were not being exposed to PFOS, fish with BPA epimutations had a lower rate of survival. Therefore, following early development, fish with BPA epimutations had higher rates of mortality in the lowest concentration. So why would fish with BPA epimutations survive at a higher rate during early development, but then have an increased rate of mortality after early development? Well, one possible explanation is that fish in this concentration also showed significantly higher rates of maldevelopment. Therefore, this could begin to hinder the fish as they grew. There were 4 distinct incorrect physical developments, also known as teratogenic phenotypes, that were observed in the fish. Fish in all concentrations showed phenotypes of residual yolk, non-inflated swim bladder, spinal curvature, and pericardial edemas. In the 0.002 milligram per liter concentration, we saw that fish with BPA epimutations had significantly higher rates of occurrence for all of these phenotypes. This increase in maldevelopment rate signifies a relationship between a history of BPA exposure and the increased likelihood of early developmental defects following exposure to novel chemicals, specifically PFOS. The present results suggest that individuals with history of ancestral BPA exposure are at an increased risk of developing adverse health effects when exposed to PFOS. This link is essential to make because now we know that the history of exposure from past generations will influence how sensitive following generations are to new chemicals. This new information can now be provided to regulatory agencies to influence a change in the regulations currently in place for these new chemicals. Establishing this relationship will also allow us to begin to move forward with new research that will begin to explore solutions to minimize the effects of exposure so we can provide a healthier life for current and future generations. Thank you for listening.